So, welcome to our Academy of Kidney Cancer Investigators lecture series. Mike Atkins is here today. I've known Mike for, gosh, at least 20 years, working together in kidney cancer. We don't always agree, but there are a few people whose opinion I respect more about kidney cancer than Mike. So we've worked together for a long time, and I thought he'd be a perfect person to talk about kind of this translational interface, this, as the title you see in the title, Bench to Bedside, not only in kidney cancer, but he's a leader in melanoma as well, which I always find fascinating, and parallels to kidney cancer. We probably don't learn as much from our melanoma colleagues as we can. So, Mike, really appreciate you joining. I'm going to turn it over to you to start, and encourage people to interrupt with questions, and we'll try to leave some time at the end to do so as well.
Great.
So thanks for coming. The floor is yours.
Thanks, Brian. I'm excited to be here. It gave me a chance to retool a talk that I often give to our fellows and junior faculty who are interested in doing translational research, but either don't have a lab or have a lab but want their research to be more clinically relevant. And so I title this Bedside to Bench Translational Immunotherapy Research. And I'll give some examples in melanoma and some of the stuff we've done, which people in the academy may not know about, and how that changes how I think about kidney cancer. And then I'll give some of the stuff we've done in kidney cancer. So these are my disclosures, potential conflicts of interest. And start with the keys to bedside to bench translational research. This is how one builds a career in this space. You need to focus on a disease, establish a cohesive multidisciplinary team, provide expert care that's not possible in the community, give a reason for patients to drive by all those community oncologists to come to you, learn from each other about all the aspects of the disease, and build a robust clinical volume to be used to address key clinical questions. And then identify at least one scientific collaborator, spend time in the lab to understand their research techniques and what is possible. And then identify some key clinical and associated laboratory questions that could be addressed within your patient population and by tests performed in your colleague's lab. Write a protocol to address the question, and then write a grant to fund the laboratory questions. So here's a sort of a description of the late stage bidirectional translational research and what's seen in bench to bedside, where you need a validated, promising target, a drug that hits the target, and a disease or situation where testing that drug is relevant. In contrast, for bedside to bench, you need a drug with efficacy, an important question about how best to use that agent, and a laboratory or pop-sci component. So for me, the best example of this distinction came when I was a member of the internal advisory committee for the Harvard lung cancer spore. And there was at the same spore meeting, internal and external advisory board meeting, there was research presented from two different categories of EGFR. So the first was genotype to phenotype, where Matt Meyerson and Pasiani at the DFCI were studying the kinome of lung cancer patients looking for mutations, and they found EGFR mutations. And then at the same meeting, Dan Haber and Tom Lynch were at MGH studying EGFR in non-small cell lung cancer patient exceptional responders versus non-responders to EGFR therapy. And they also found EGFR mutations in those exceptional responders. So I looked at that and said, gee, this phenotype to genotype research actually has more value, because you know already that you have a leg up in proving the relevance of your finding if you go from phenotype to genotype. So in order to do a translational investigator-initiated protocol, you need this important question. you need a cutting edge tool to address the question. And then you got to build a table, which is access to agents, appropriate patient population, regulatory approvals, and funding for the trial and correlative research. And you need all the legs of this table at the same time in order to be able to conduct your research. And unfortunately, there's also this gauntlet that you have to go through of approvals for each of these steps, which gets harder and harder despite the efforts to make it easier and easier to get things approved to actually run your trial. So you have to build the table strong enough to support all the paperwork that you have to do in order to get these approvals to get the trial launched. And I know this is ringing true for Brian, who's gone through this step many times. And I know that for my melanoma trial that was finally published, I had 23 amendments after the study was approved before it could actually be completed. So that's just, a lot of work to get it approved. And once it's approved, you got to keep going. But if you do this, you're an expert in this particular question. And I tell my fellows and junior staff, do not sell yourself or your data short. It's very valuable. Don't just hand it to your colleagues. You have to be part of how it's being used. And because it's unique data that's very valuable. So I'm going to focus translational research on cancer immunotherapy. And I'd like to start this by highlighting what the principles of cancer immunotherapy are that distinguish it from tumor-targeted or tumor microenvironment targeted therapy. So as Jed Walchuk had said, with cancer immunotherapy, you're treating the immune system so that it can treat the cancer. And because the activated immune system can target many tumor antigens simultaneously and deepen and broaden over time, immunotherapy can cure patients with metastatic cancer. It may be the only thing that can do that. And therefore, the hallmark of effective immunotherapy are plateaus on the KM curves, particularly plateaus that exist once treatment has stopped. And so if you're a true immunotherapist, you're looking for those signs as you look at the Kaplan-Meier curves. So what are some examples of translational immunotherapy questions that you could apply to any disease? How to sequence and combine with standard of care, predictive biomarkers for response, mechanisms of resistance, how to treat anti-PD-1 resistant disease, identifying the right combination for a particular patient, when to stop therapy, and how to predict and best treat immune-related adverse events. So I'm going to go through some of the research we've done studying these type of questions. First in melanoma, looking at the DreamSeq trial, the studies we've done with serial biopsies in BRAF mutant patients getting targeted therapies, and then a PET stop trial trying to figure out when it's safe and to identify a biomarker for stopping treatment. And then look at some renal cancer translational studies that are similar, and I'll get to those. So in 2015, for the treatment of BRAF mutant melanoma, we had evidence of progress. We knew that BRAF MEK inhibitors could raise the overall survival bar and tend to splay the curve, and that immunotherapy, particularly combination Nivo-Ipi, could raise the tail. And so the question we asked, which is not that different from the question we're asking in kidney cancer, is which is preferred? And because patients would have access to both of these treatments, is there an optimal sequence? So to address this question, we launched the DreamSeq trial, which took patients with metastatic BRAF mutant melanoma, stratified them by some key risk factors, and randomized them to either Nivo-Ipi induction followed by Nivo maintenance, or dabrafenib trametinib continuously. And if there was disease progression, they crossed over to the alternative therapy, assuming they met the eligibility criteria. And we chose as our primary endpoint 2-year overall survival, and secondary endpoints are shown here as well. And this study was stopped early by the Data Safety Monitoring Committee when 59% of patients had reached 2 years of enrollment because of a clinically meaningful difference in 2-year overall survival. So this is the overall survival curves, and you can see that they were biphasic. They crossed but by the time you got to 2 years, there were 100 patients that had died, 62 on the Dabtram first arm, 38 on the Nivo-Ipi first arm, and there was a 20% difference in 2-year overall survival, which was statistically significant. And the 3-year overall survival difference was even greater, 24% difference. And so the question we asked then was, why is IO first better? And can we identify a population to get targeted therapy first? So one of the reasons why IO was better was, even though first-line response rates were similar, and for targeted therapy, second-line response to targeted therapy was similar to the first-line response, immunotherapy in the second line appeared to be less active. And this was true for PFS as well, where the PFS was slightly better for immune therapy first. It was similar for targeted therapy, whether given in the first line or the second line, but immunotherapy in the second line, if you waited till patients had progressed on targeted therapy, produced a very poor PFS. Another reason why immune therapy was better was that the duration of response, even though response rates were similar, the duration of response was significantly better, with 88% of patients responding to immunotherapy still being in response, while half the patients responding to targeted therapy had progressed. And so a major significant difference. And then one of the final reasons why immunotherapy was better and why the duration of response was better was that more patients, as shown here in red, tended to relapse in the CNS with targeted therapy, while relapse in the CNS was unusual with immunotherapy in patients where their systemic disease was controlled. And so we know from our other studies with Ipi, Nivo, and melanoma that it works as well in the CNS in patients with asymptomatic metastatic disease, or in this case, it was patients who had non-detectable or treated metastatic disease who were on the study. So another thing that we looked at in trying to identify who should get targeted therapy first was that for every risk factor that we looked at, and that shouldn't say age, it should say gender at the left-hand side, the blue line, which is immunotherapy first, was above the orange line, which is targeted therapy first. And that was true for patients who were deemed to be the patients who responded best to targeted therapy, meaning those with a low LDH and low disease burden, M1A or M1B disease. And also LDH, which was a known risk factor, one could see that LDH, no matter what your LDH level, you did better with immune therapy first. And that's just shown graphically here, where if your LDH is normal, the curves separate earlier for both PFS and overall survival, as you can see here. But even with the LDH is elevated and the PFS is lower and the overall survival is slightly lower, you can see at some point by 2 years, the curves are separating and your long-term survival is better if you got immunotherapy first. And so we've been doing a lot of research on the tumor tissue. There were a lot of correlatives that attached to this study. And we had 5 grants that were funded to do correlatives. Many of those expired before the study was actually completed, which was a lesson we learned to not apply for correlative funding too early. But one where we still had money and we could still look at was looking at tumor tissue for predictive biomarkers. And I'm going to present a lot of data at SMR about this. But one thing that I thought was relevant in today's talk was looking at the angiogenesis signature and impact on progression-free survival. And you can see that patients who had angiogenesis high, which is the lighter blue curve here, had worse outcome to immunotherapy than to those with low angiogenesis signature. And that didn't seem to have as much impact on targeted therapy. So this, I think, has been, in my view, erroneously interpreted as the factors associated with high angiogenesis signature are immunosuppressive, which I don't think is actually the case. I think what actually it means is that tumors that are hypoxic respond less well to immunotherapy, which is why the angiogenesis signature is high. And we'll come back to that discussion when we talk about how it applies to kidney cancer.
Mike, can I interrupt real quick on that one?
Sure.
Just to go back.
Sure.
And just, you may have said this, how was the angio signature? Was this RNA-seq? Was it?
That's RNA-seq, yes.
Okay.
Yes.
I mean, again, at least in kidney cancer, I think just the angiogenic and immune responsive phenotypes are just a little bit different, right? I'm sure there's overlap. So your angio low might just be your immune responsive. Is that, what do you think of that?
No, I think in kidney cancer, the angio high are the immune responsive.
Okay.
Because kidney cancer is different because it's not hypoxic when angio is high.
Yeah. Okay.
So that's where I think this whole discussion, and I'll get to this, you know, all the beautiful work that we did with emotion 150 and 151 missed the point.
I'm looking forward to
it. Okay.
Very good.
So in looking at the DREAM-seq clinical conclusions, I think these are practice-changing results. They will improve patient overall survival if you give immunotherapy first. And the results are potentially relevant for other diseases if you're talking about how to sequence therapies. And the correlative studies will, I think, identify subsets who do better with either approach, although it may be hard to find a group that should do better than targeted therapy, although I've done my best and we'll try to present that at SMR in a few weeks. So another thing that we've looked at is triple combinations, giving BRAF MEK inhibitors with anti-PD-1. And so there was data that came out about a decade ago that suggested that treatment with a BRAF inhibitor results in increased melanoma antigens and CD8 T cells in tumors of patients with metastatic melanoma. And this was associated with increased markers of T cell cytotoxicity and a decrease in immunosuppressive cytokines and VEGF, but an increase in immunomodulatory molecules such as PD-1, TIM3, and PD-L1. So this all supported the hypothesis of potential synergy of BRAF targeted therapy with immunotherapy. However, there were important questions that remained. And one of the questions we wanted to know was this increase in immune cells in the tumor microenvironment, was it real or was it an artifact? And so in some preclinical studies that were done with Nick Restifo and Nick Aquavella, who was a fellow working with us at that time, we showed that in this mouse model that the immune cells clearly increased in a patient who was getting the BRAF inhibitor, vemurafenib. However, if you looked at tumor size, you could see that the tumor size had decreased significantly as well. And so if you actually looked at the total immune cells that were in the tumor at the time when you were doing that second biopsy 10 days later, there was actually no difference. You killed off the tumor cells, but you did not increase or even change the immune cell population. So then we did this study where we did serial biopsies in patients with BRAF mutant melanoma. We did a baseline biopsy. We did a biopsy at 8 days, 15 days, and 28 days into treatment. And what we saw was there was increase in immune cells, CD8 cells here in the at 8 days, and you could even see these tertiary lymphoid structures appearing to form at 15 days. And at 28 days, there was a lot of tumor necrosis going on, which is this background. But it looked like there was an increase in CD8 T cells, similar to what we saw in the mouse models. However, there was also a similar increase in CD4 T cells and in FOXP3 T cells by day 15. And so this was not something that is restricted to cytotoxic T cells that might have been there recognizing the tumor. And if we looked at the CD4 to CD8 ratio of the TILs, it didn't change at all in the patients on our study. So we did a lot of other work on these serial biopsies, which I don't have time to show you, but I'm going to summarize by saying that the any TCR V beta clonal expansion tended to diminish after day 15. There was no significant increase in proportion of antigen experienced or activated T cells as measured by interferon gamma or granzyme B expression. Some showed some increase in PD-L1 expression relative to SOX10, but this was not required for treatment efficacy because all the patients showed some tumor shrinkage. And our conclusion was that BRAF treatment is not associated with the generation of new and or specific tumor immunity. In summary, that meant that BRAF MEK inhibitor was not an immune therapy and was unlikely to synergize with checkpoint inhibitors. So this was our data. Despite this data, the pharmaceutical company and investigators could not resist looking at triplet studies of BRAF MEK inhibitors plus an anti-PD-1 versus BRAF MEK inhibitors. There were 3 studies done. They all had similar results, although one was positive, just slightly positive, and led to FDA approval of Vemcobi plus atezolizumab. But if you look at these curves, you can see for PFS, there's really not a dramatic difference for the triplets versus the doublets. And if you look out at where the tails are of these curves, keeping in mind that the patients who are progression-free on targeted therapy are still getting targeted therapy, they're about where Nivo/Ipi was at 5 years for progression-free survival. And I would point out that the patients who are progression-free on Nivo/Ipi, if they are all off treatment and if they progressed, could have gotten a BRAF inhibitor and still responded to it. So people don't use triplet therapy because of that information in melanoma. So when to stop therapy? So there's data that showed for patients who had stopped therapy because of toxicity by week 12, that the response rate was at least as high as that of patients who didn't have toxicity in melanoma, and that 64% of those patients had ongoing responses. So what we did at Georgetown was see if we could identify a biomarker for those patients who didn't have toxicity, weren't forced to stop treatment, that could tell us it was okay to stop treatment. So what we did was take patients who were tolerating therapy and were having either stable disease or response, partial response or complete response at a year, and subject them to a PET scan. And if the PET scan was negative, they were metabolic CR, and that encouraged us to stop treatment. If the PET scan was positive, we did a biopsy, and for half of those patients, when we biopsied them, we found no viable tumor. We found melanophages, which are macrophages stuffed with melanin. in the place where the PET was positive. And the PET was positive because there were probably still immune cells there trying to clear up what was a dead tumor. And when we looked at how those patients did when we stopped treatment based on these reasons, they did at least as well as our patients who stopped treatment due to toxicity. And only one patient had progressed, and that patient progressed with an isolated axillary lymph node that was resected, and none of these patients have required additional systemic therapy. So that actually turned my melanoma clinic into, from a sad place to a virtual travel agency where patients unleashed from their treatment were free to travel the world, and they would come back talking about where they've been and where they're going. And I was left to talk to them about my experience in those places and what I knew about the place they were going and the things that they should do. In short, we turned these survivors into thrivers who were back to their precancer lives, albeit wiser and with more gusto. So we wanted to see whether our experience could be broadened. And so my colleague, Jeff Gibney, launched the PET Stop study, which was applying this experience in the cooperative group and our approach to a broader group of patients. The only difference is that patients who had positive biopsies or where you couldn't easily biopsy their disease would continue on therapy for another year, and then we would repeat the process. So this study is busy accruing in the cooperative groups, and hopefully will prove that this is an approach that's worthwhile. We're also looking at PET readouts with ECOG-ACRIN, the biopsy processing with getting support from BMS, and we're going to work with SITC Sparkathon team to analyze blood for ctDNA and Natera assays and the tissue for biomarkers of resistance. And so future, I think we're going to try to see if we can shorten this to less than a year using imaging and or biopsies and or ctDNA or the Natera approach, and see if we could expand this approach to other cancers such as kidney cancer. So I'm now going to move on to discussing some of the things we've done in kidney cancer, and I'm going to focus on some of our old studies about mechanisms of resistance to VEGF receptor TKIs and how that might apply to the IO TKI data, then some IO biomarker studies, and then talk about some new clinical trials, particularly the Architect study that we're currently running. So in some of our early mouse studies looking at sunitinib and sorafenib, we saw that using arterial spin-labeled MRI, which is a measure of tissue infusion or perfusion, that when you give, this is a xenograft of a kidney tumor line here in a mouse showing all this perfusion. And when you give a TKI, within 3 days, all that perfusion is gone. You actually have an infarct developing in the tumor microenvironment, and you lose all your blood vessels that were present before. That's within 3 days. By day 9, you're getting this resistance developing, even though the tumor hasn't grown, with this perfusion developing on the outside, this rim of perfusion here with the infarction on the inside and some blood vessels performing. And by day 22, when the tumor is growing again, you can see the perfusion has restored. So this is a mildly sensitive kidney cancer model. But if you look at the 8498 model, which I think behaves more like human kidney cancer, you can see that that resistance takes a little bit longer. By week 1, the perfusion is reduced. By 4 weeks, still reduced, continued to be reduced all the way up to week 6. But you're starting to see perfusion coming back at week 7, even though the tumor has yet to grow. And then by week 10, the perfusion is increasing if you look at these numbers, and the tumor is starting to grow. And by week 12, perfusion is back to where it was at baseline, and the tumor is resistant. So how does this apply to humans? So here's an example of a patient getting sunitinib with a MRI at baseline, showing this tumor with perfusion here, perfusion gone at week 2, if you can see this red circle here. And by the beginning of cycle 2, or about week 12, the perfusion is coming back. So similar findings in humans. In the study that we did, you can see that perfusion goes down by 2 weeks, still down at cycle 2, still down at cycle 4. By the time of progression, the perfusion has come back. So perfusion in humans, when you get TKI therapy, is a measure of angiogenic escape, and resistance to TKI therapy is by upregulation of angiogenesis, more than just VEGF. It's things besides VEGF that cause that escape. And so because the tumors prefer to grow with VEGF, if you stop the TKI, give them a little bit of a drug holiday, and take that resistant tumor and implant it into another mouse, which is an example of doing the same thing, that tumor is once again sensitive to sorafenib. Now, this is not very sensitive. This is a 786, which was resistant by 10 days. But you can see you get this plateau again in tumor growth when you restart the sorafenib. And in order to create a longer response, you need to use an agent that blocks multiple mechanisms, not just VEGF of the angiogenic escape. And so this is just looking at imaging in those patients. You can see that with that restarting of sorafenib, you can once again see this loss of perfusion, decrease of perfusion, which is associated with necrosis. And by the time you stop, the angiogenesis has been restored. So if you look here, this is the angiogenesis at baseline. You restart, this is necrosis at the time it's restarted, and then when it stopped, the necrosis is gone. And we then looked at whether you can slow growth by giving treatment intermittently, giving this drug holiday to keep the tumor sensitive to the VEGF inhibitor. And what we saw is that if you gave a standard dose treatment continuously, you saw better inhibition than intermittent. However, if you gave the same amount of therapy at a higher dose, but let's say over 3 days instead of dividing it over 7 days, you can see that you delayed progression compared to giving that same dose at a lower dose continuously. So this is an example of what we saw in that study of looking at sunitinib 37.5 continuously versus 4 weeks on, 2 weeks off of 50. And so you could see that in the mouse model. And you could see also that there were changes in perfusion when you gave the dose continuously. You could see that perfusion came down, but eventually you saw an escape, and the perfusion came back by 10 days post-treatment. While if you gave it intermittently, you could see the perfusion came down quicker at day 3 when you were giving this higher dose, but by day 5 or day 7 when you were off treatment, it had come back, but it came down again when you restarted. And overall, there was lower tumor perfusion at day 10, giving a higher dose intermittently than continuously. And that's just shown graphically here. If you compare the blue bars and the orange bars, where the intermittent therapy leaves you with lower perfusion at day 10. So if looking at the implications of all these perfusion studies with the VEGF receptor TKIs, you can see that changes in perfusion may be a predictor of response to the VEGF pathway therapy. The higher perfusing tumors, which I didn't show you, actually are the ones that respond the best to the TKIs. And tumor perfusion can be a surrogate of tumor viability. Restoration of perfusion can herald the development of resistance, and perfusion studies in animal models can help direct translational research in clinical care, support for retreatment, explore schedule issues, identify mechanisms of action of agents, identify effective combinations, and identify and credential new therapeutic targets. And could also be looked at, although we have never done this because I moved away from Boston and there's no immune competent VHL null kidney cancer model, but could be looked at in humans in IO TKI combos. So that brings us to the discussion of what should we do in the front line, and we have IO TKIs or anti-PD-1 CTLA-4 as options.
Can I interrupt again real quick?
Sure, of course.
I mean, I'm a big fan of breaks on TKIs.
Yeah.
There's some studies that are trying to look at it, but it's pretty hard to look at it formally. There was that TIDE study presented at ESMO. I'm not sure if you saw it. But I wonder if it doesn't even have to be formal breaks at 9 months, this formalized long on, long off. And I just tell people take 3 to 4 days off whenever you want, like shorter breaks around life events from a quality of life standpoint is probably more impactful.
Yeah, I do the same. And if someone has, let's say, toxicity, let's say with Cabo, where it's rather than reducing the dose from 60 to 40, I might try, say taking it 5 days out of 7 or something like that. Yeah. Or if they're at 40 and I don't want to go down to 20, I'm even more likely to tell them to do it more intermittently. With axitinib, I say, if you feel there's a problem because the half-life is so short, take 2 or 3 days off and it should recover.
Yep. I guess my question from a chronic hypoxia being bad resistance mechanism, do you think those more frequent shorter breaks or less frequent longer breaks would be better?
Yeah. Well, I think, the issue with hypoxia is, completely different if you're trying to give immune therapy with it. And I don't know that you ever restore fully if you're taking a break and then you give the TKI again, you're making the tumor hypoxic again, which is good for the TKI efficacy, but it may not be good for the IO efficacy. So it's complicated.
Yeah. Okay. Just curious. Thanks.
Yeah. So I'm going to go to this rationale for combining checkpoint inhibitors with VEGF inhibitors, which everybody cites. And there's a lot of preclinical data. You see all these references at the bottom to support all these different steps for how blocking VEGF might improve the immune response, like promotion of T cell priming and activation via dendritic cell maturation, normalization of tumor vasculature and increased T cell tumor infiltration, and establishing immune permissive tumor microenvironment by decreasing MDSCs and Tregs. And so maybe that's true in all these preclinical mouse models, but none of these models are done with VHL null immune competent kidney cancer. So I don't think they apply to kidney cancer where these tumors are not hypoxic to begin with. And so when I look at, Brian's study here, which, by the way, I did participate in, and I did actually develop the Axi-Pembro regimen in the phase 1, I'm looking to see whether the curve is behaving like a TKI curve or an immunotherapy curve. And so I just follow how the curves go as they go down. And particularly, for the favorable risk population down here in the left corner, I see a curve that just goes down the way a TKI goes down. And I think when you're looking at duration of response, it's not a duration of response curve that we typically see with immunotherapy. Now, that may be because they're more responders, because there's also a TKI response there. But one has to, consider the fact that the responses that you get are not all due to the immune therapy, and that you could have gotten those responses potentially in the second line in the patients didn't respond to the immune therapy. And so same thing with the CLEAR study. In a CLEAR study, the curves are more separate, but when you look at overall survival, the fact that those curves have the same median overall survival between len-pembro and sunitinib is just disturbing. And to me, we were arguing about this a little earlier, but that just means you just can't give the same therapies after len-pembro that you can do after sunitinib to impact overall survival and expect the same benefit. And contrast that between the nivo-ipi in Checkmate 204 versus cabo-nivo in 9ER. You can see that cabo-nivo at 3 years already has a plateau, if there is one, in their PFS curve that's below what we see with nivo-ipi, even though the sunitinib curves look essentially identical. And so people will point out this box that these are people who aren't benefiting from nivo-ipi. And I say that's true, but they can still get cabo, and we know from the CONTACT-3 study that they can benefit pretty well from cabo with a 40% overall response rate. And if you look at putting the PFS curves together, as Tony Choueiri did, you can see the sort of the highest PFS curves go with the more potent TKIs. The highest one on top is the pembrolizumab, while the atezo-bev in red is down at the bottom. But you can see the curve out here with the longest plateau and the longest follow-up is the nivo-ipi. And once again, these patients can still get a TKI and get all the benefit of a TKI. And it's also highlighted in this comparison that Meredith Reagan did of the hazard ratios, where you can see for 214, the hazard ratio for overall survival up here in this right hand is a plateau, which is maintained, while for CLEAR and for 9ER, and for Keynote 426, these hazard ratios are tending to go up over time, indicating that even though the PFS benefit is real and sustained, that the overall survival benefit is not as sustained over time because people on sunitinib have access to more effective therapies. And this is particularly also true in the favorable risk population, where all the hazard ratios over time are leveling off at around 1 compared to sunitinib. And I just call your attention to this group here in Checkmate 214, which is the reason we can't give pure immunotherapy to favorable risk patients. And I'm not going to go into that in detail, but I think there was probably some issues with what patients got after they got immune therapy in 214, while that explains why their overall survival was not as good as it could have been. And I don't think it's relevant to 2023. So this is Brian's data. Worked with this data in the Emotion 151 study, where you can see these 7 clusters, which are, I think, really important clusters, which show the angiogenic group and the T effector proliferative groups that appear to do better with sunitinib in the angiogenesis group appeared to do better with atezo-bev in the proliferative group. And I think this is an important slide when you look at what makes up the favorable, intermediate, and poor risk populations by these IMDC categories. And you can see that for the favorable risk, somewhere around, I think 50% or more are driven by angiogenesis, and then there's another 25% that are complement, and only a small percentage of those have this, what would be immune therapy responsive disease. And so, I think it's reasonable to say if the angiogenesis does best with TKIs, the TKIs could be given here. But how do we explain the fact that the response rate to immune therapy is similar in this favorable risk group, and there's a plateau on the PFS curve that's in a similar place to the other categories? And the duration of response is also pretty good in this favorable risk population. And when you look at TFS, treatment-free survival, the favorable risk group has the longest TFS over a 60-month period compared to the intermediate and poor risk. And with IO TKIs in the abstract submitted with ASCO, the TFS is not any different than sunitinib. And so, to me, this suggests that the angiogenic group is not the same as the angiogenic group in melanoma that responds poorly to immunotherapy because they're hypoxic. Kidney cancer is different because it's not hypoxic until we give them the TKI. So, question is that we wanted to ask in our studies was, is there a regimen for favorable risk patients? Can we identify responders to nivo monotherapy? And can we identify biomarkers of resistance? So we launched the HCRN GU16260 study, which gave a biopsy for patients, then gave them nivo. And if they were PR or CR, they continued nivo monotherapy to 96 weeks. And if they were non-responders all the way out to 48 weeks, they had another biopsy and then went on to boost nivo-ipi. So if you look at first line, you can see that we had, on the waterfall plots, a fair number of favorable risk patients respond. Overall response rate was 34%, but the favorable risk patients had a 57% response rate, while only 25% in the intermediate and poor risk patients. And these responses were meaningful. The median duration of response overall was about 19.3 months. And for the favorable risk patients, 17 out of 20 responders were ongoing. And if we wanted to look for biomarkers to see if we could identify who responded, we saw that PD-L1 was actually a decent biomarker for predicting a response. If the PD-L1 was really high, 75% of patients responded. The problem was that 27% of patients responded even if the PD-L1 was low. So here are the waterfall plots that go along with the slide I had just shown, and you can see the high degree of tumor shrinkage in the PD-L1 high. The problem was that represented too few percentage of patients, and therefore couldn't be used as a biomarker, but could be a component of a biomarker. But what we showed with this study was that all the other studies, I think, with PD-L1 as a biomarker were confounded because the PD-L1 analysis was done on the primary rather than a metastatic lesion as we did it, using samples that were collected more than 12 months before treatment initiation, and often before an intervening treatment, which tended to change things, looking at time to treatment endpoints such as PFS or OS that are poor surrogates for IO efficacy as compared to overall response rate or landmark survival also was a problem, and using combination regimens that don't isolate the impact of anti-PD-L1 was also an issue. And sometimes comparing results to a control arm rather than within the ICI treatment arm was also an issue when looking for biomarkers. And so,
Mike, can I ask a question?
Sure, of course.
The results in favorable risk in your study are notable for sure, but if you look at like Keynote 427 with Pembro, they're not nearly as good.
Yes, but we have not seen the long-term data with Pembro in the favorable risk population.
Well, maybe not the long-term data, but I think we know response rate and CR. I don't remember the difference. Actually, that median follow-up was almost 36 months.
If you look at the PFS tail in the favorable risk population, it's actually quite good. It's just not been published.
Okay. I mean, I know response rate is.
I think there's a reason why it's not being published.
If you look at response rates, which I understand are imperfect and maybe not the perfect immunotherapy endpoint.
Yeah, the response rate, I think it's fair to say that this is an outlier. The overall response rate with Nivo was similar to what we saw in Keynote 427. It's just where the responses happened. Yeah. And so I think the point is that it's not that favorable risk patients are more responsive. It's just that favorable risk patients are not less responsive.
Yeah, I guess I don't disagree. My question was that maybe your subset of 35 favorable risk patients happen to be more of the cluster 4, 5, 7, and those in 426 happen to be more cluster 1 and 2, right? Because we know it all exists in each IMDC category. So
and
I don't know if you have data.
It's certainly possible, and that data will be coming out soon. But I'm going to move along because I've got short time and I want to cover a few things. So some of the biomarkers we've looked at are looking at SLAM-F7 through single cell RNA-seq and at PD-1 positive Treg numbers based on data from Sabina Signoretti and Arlene Sharp. And so looking at SLAM-F7 in this study, we saw that patients who had high levels of SLAM-F7 on their CD8 T cells were more likely to have progressive disease, and PFS and overall survival was worse for that population. Small numbers, but something worth considering. When we took that to Checkmate 09 and Checkmate 025, you can see that more patients had progressive disease if they had high SLAM-F7 positive T cells, and there was a slight difference that was statistically significant in overall survival. We also wanted to look at CD8 positive TILs expressing PD-1, but not TIM3 or LAG3. And that was looked at in Checkmate 10 and Checkmate 025. And we found that these non-terminally exhausted PD-1 positive CD8 T cells were associated with better outcome, both IR-PFS, and when you combined it with PD-L1 positive tumor cells, also better outcome than those that were negative. And the same thing was true in the nivolumab arm, looking in the second line in Checkmate 025. And so we wanted to apply that to the HCRN data, and we saw something similar in an abstract that was at ASCO this year. The non-terminally exhausted PD-1 positive CD8s were associated with better outcome, and that was even better when combined with PD-L1 positive tumor cells. So the implications of this data are that this is a rationale for looking at combining anti-PD-1 maybe with anti-CTLA-4 and an anti-LAG3, and Brian is leading trying to develop a study within the KCRS consortium looking at a triplet with a LAG3 compared to Nivo-Ipi with Rela and also potentially with Regeneron. And I think those things are worth doing based on this data. We also saw, and this is interesting data, that PD-1 positive Tregs were associated with worse outcome, PFS, and overall survival in the 025 studies. And to us, this just supported Arlene Sharp's data, where if you have PD-1 positive Tregs and get anti-PD-1, it makes the tumor more immunosuppressive. And so this suggested that there's actually a balance in the tumor microenvironment between PD-1 positive CD8s and PD-1 positive Tregs. And the higher the PD-1 positive Tregs in that ratio, the more resistant to PD-1 therapy you're going to be. And the higher the CD8 positive PD-1 in the ratio, the more responsive you're going to be. So we wanted to see if we could identify things that could eliminate PD-1 positive Tregs. And so we went to the Agenus drug botensilimab, which has this FC enhanced region and also seems to deplete Tregs and has activity in a fair number of diseases as a single agent and in combination, which nivo-ipi does not, including MSS colon cancer, where there was 25% response rate and most of those responses being durable. And the toxicity profile looked to potentially be more favorable than what we saw with nivo-ipi. And that encouraged us to launch the Architect trial, which is comparing this botensilimab and balstilimab to nivo-ipi in treatment-naive patients with clear cell kidney cancer. And we're submitted a DoD grant now that the study's open and accruing within the HCRN, looking at if we could identify a difference in response in the patients with PD-1 positive Tregs with the BOT/BAL compared to nivo-ipi, or using that ratio. And we'll also use it to look at things such as SLAM-F7 and quantitative immunofluorescence for PD-L1 expression, as well as looking at other things such as these signatures from emotion 151, sarcomatoid features, and potentially next generation sequencing. So I'm going to close by saying our goal when we're treating kidney cancer, as it should be, most cancers should not be turning cancer into a chronic disease, but we should strive to make cancer a curable disease. And in order to do so, I think using agents and combinations at first line that maximize the anti-tumor immune response is critical to achieving that goal. And I want to stop there and just thank and acknowledge my collaborators at Georgetown within the Dana-Farber Harvard Kidney Cancer Program, other collaborators in both melanoma and kidney cancer from around the country, and my funding sources. So thank you very much. I apologize for going all the way to the end of time.
You're good. You're good. That was amazing, Mike. Thanks. I really appreciated it. Maybe if we have burning questions, if people still have time, I'm going to have to jump off soon, but there's a lot to ask here. I feel like we can do a whole session on it, but if anybody has a question or comment.
I have kind of curious about the fact that that how does this one HIF1 status can impact the response to immunotherapy?
The HIF status? Yeah.
Yeah, the HIF1 status, like there are so many occasions where 14q deletions are there. So I just want to know that isn't it?
Yeah, well, I think there are obviously two things that are going on with immunotherapy response. I think one is immune competence related to normoxia in the tumor microenvironment is critical for having an immune response. But I also think that there's some of these more aggressive tumors that are in the proliferative phase and they're more inflamed, likely have more antigen expression. And that may be related to HIF2 alpha and HIF1 deletion. And I think HIF1 expression, if it's still there, may, I sort of have this thought that HIF1 is deleted because it doesn't work as well in terms of driving angiogenesis in a normoxic environment as HIF2. And so if HIF1 is still there, it probably goes along with more hypoxia and probably less response, but I think it's something that needs to be looked at.
Thanks, Mike. This was great. I think we'll jump off
Thank you.
for time, but appreciate your time today. Look forward to catching up on all this soon.
Yeah, I'll see you next weekend. Yeah. All right. Thanks again. Bye-bye.
All right. Thanks all.